describe it on the
Hello everyone, welcome. I'm glad you could all be here. So, let's see. Portland, just day of learning. All right. The agenda for tonight, this evening, I'll give a quick little speech, and then of course I'll have my awesome dad talk about the treatment updates and everything that's going on with that. Then we will have Dr. Christopher Corless, who will be referring to pathology and mutational testing and more scientific stuff. Then we'll have Tracy Walker come up and referring to like finding and participating in just trials, clinical trials, and yeah. Then Life Raft group programs and services with Michelle. And after that, we will have quick questions and answers. So the background of this event. So for this, first of all, this is like a senior project, and it's involving more independent and more in-depth by more, let's say it, more in depth of trying to be more successful and thinking of a way of your passion of doing something you love and helping others. For example, other people have done lots of different projects regarding helping the homeless, other stuff like that. Mostly something around the community. I'm specifically. The reason why I picked GIST is one, my dad. Obviously, he's considered an expert, and he's when he works off of this, when he comes home every night, I'm always curious and asking him questions. I've actually been to 3 different events. 2 were fundraising walks, and that was probably the second fundraising walk was probably my favorite. One of my favorite people, Martha, she's one of the patients, and she's been, like, I don't know, like I just kind of bonded with her, and it's really nice to meet her, and she's always entertaining. But I don't know, like just experience from talking with them, and and having my dad already, know about it, and that's like his life, I started getting more involved with understanding how his life outside of the house is. Quick picture, Martha is the one on the right, and she's like my favorite. So what is Just Day of Learning? It's a free one-day event to help patients, caregivers learn more about the specific cancer, and I guess just get more of a knowledge for it. And I guess a way to support and navigate their way, through the journey of it. Obviously, as I said again, this whole event will involve my dad speaking, Chris, Michelle, and Tracy. But yeah. And then this is at the second walk of me and my dad. So yeah. And then introduce my dad, Dr. Heinrich.
Thanks, Mark. I have to find my presentation. Yep, that's Michelle. Okay. Okay, so first I wanted to thank a few people. So Michelle, Ellie, and Ashley have been responsible for arranging everything. So thanks to them for sending out all the invitations and setting everything up. Also, I wanted to acknowledge members of my clinical team, research team. I see if you could.
No.
Sorry. As members of the clinical team, I see Tracy, Diana, and Lindsay, if you could stand up. So some of you. They're the ones that if you're in a clinical study or trying to get in a clinical study, they're the ones that are helping you navigate that, and Tracy will talk more about that experience. I also see some of the people from my lab who do the research that leads to the new drugs, so if they could stand as well. Okay, so this is going to be just an update on treatment, so we can talk about research things or whatever at the end, but this is kind of more straightforward, keeping with the day of learning, teaching you about your disease. So the things we'll talk about a little bit about just background, definition, a little bit about the biology, although mostly just introducing what Chris is going to talk about. And basically one of the messages I hope you take away from today is mutation, mutation, mutation. So knowing what's wrong in your tumor is very critical to being able to treat you in the best possible way. And fortunately, we have world-class, through Dr. Corless testing here, but a lot of patients, probably 90% of patients with just in the United States, don't know what the mutation testing is, and that leads to them not receiving always the optimal care. We'll talk about the role of surgery and what we can sometimes do if your tumor is too big to be removed surgically. There's some things we can do. If we remove it successfully, consideration of should we give you something after your surgery to keep it from coming back. That's called adjuvant therapy, so we'll talk about that. And then in cases where the tumor is spread to distant spots from where it started, we'll talk about how that's treated, both the first-line treatment and then what happens if the first-line treatment, which is Gleevec or Imatinib, if that doesn't work, what do we do next? We'll explain or talk a little bit about why that happens, how we overcome it, and that sometimes will lead into more of what Chris is going to talk about and also what Tracy is going to talk about. So, a lot of people probably know this, but I guess we should start with what is a GI stromal tumor or a GIST? So this is a type of cancer that if you would have asked this question at the time that Mark was born, or even until he was 3 years old, there would not have been a definition of this because there was no recognition prior to the year 2000 that there was a thing called GIST. So it used to be lumped in with something called leiomyoma, or it was lumped in with leiomyosarcoma. And the reason for that is that, as Chris will tell you, mostly when we diagnose cancers, we're using the best technology of the 1800s, which is a light microscope. And so we're looking at things and saying that these things look alike or they look different and giving them names. But as we've come to realize, not everything that looks the same is the same. So not everything with 4 legs is a dog, or even a dog versus a wolf is not the same thing, even though they look pretty much the same. So there are differences that we can draw out. So prior to the year 2000, we didn't actually even realize that there was something called a GIST. But when Mark was about 1 year old, there was a very important paper published by a group from Japan. And so they first identified some important things that we still use today. One is they identified for the first time that there was something called the KIT protein, and we'll talk a little bit more about that, that was expressed in these types of cells, and that you could do a pathology test and show whether the KIT protein was there or not, and that would allow you to say that, aha, there's something new that we're going to call GIST, and these other things that it looks like, we're going to call them something different. And more importantly, for how the story went down, not just in terms of diagnosis, but in terms of treatment, they described for the first time that there were mutations of the KIT gene, which led to activation. And as we'll talk about, the KIT gene is turned on and is the power supply for about 70% of GIST, and we'll talk about the other 30%, and Dr. Corless will talk a little bit more about that. But what we learned from this really original paper was that these mutations cause the enzyme to be activated, which means really the power source for the cancer was turned on abnormally, in these cells, and we'll talk a little bit more about that. So this immediately allowed us to test for KIT expression. So what you're seeing on this slide is you can take an antibody which recognizes the protein, and then you can do an enzyme reaction that turns where the antibody and the protein are brown. So when you're looking at this, all of the tumor is brown, and the normal tissue at the top is white, and so we can say that this tumor expresses the KIT protein, and most tumors do not. So this allowed us to have a uniform and now universal way that we could look at something and say, this is a GIST, or this is not a GIST. Prior to this time, we did not have that. We also realized that even though we had a suspicion that there might be something going on with stomach tumors, that they weren't all the same, we then realized using KIT staining that these GIST tumors could arise at other locations. It had also provided a clue to where these tumors were coming from. So most stomach cancers or colon cancers start on the lining cells. They're called adenocarcinomas. They're from the lining cells. But GIST actually arise in the wall of the stomach or gut because they arise from the pacemaker cells. So a long time ago, someone who actually won the Nobel Prize for Anatomy and Physiology, Santiago Ramon Cajal, had described these cells, which have come to be called the interstitial cells of Cajal. And these are the pacemaker cells that allows the stomach to grind things up and to push things basically from one end to the other without kind of going sideways or backwards. And it turns out that GIST cells arise from these pacemaker cells, which is why they act so unique and different from most cancers of the GI tract, which arise from lining cells. So armed with this ability to diagnose GIST, and Dr. Corless will talk more about this, we realized that the most common site for GIST to arise was the stomach in about 70% of patients. The next most common site was the small intestine and 5% in the colon, but there's also oddball sites like the esophagus, the rectum, the appendix, the gallbladder, in which patients can occasionally have GIST arise from. So these things arise as a mass, and I think Dr. Corless has the same picture because I got it from him, but his is turned sideways. So basically this is showing a GIST, and this is the stomach that's been cut open after an operation, so part of the stomach's been removed, and the tumor is basically this part. It's been cut in half, so it's kind of been filleted open. And you can see in the center of it, this kind of looks like blood, and that's because the tumor outgrew some of its blood supply, so it actually kind of hemorrhaged internally. So this is 5 centimeters, which is sort of moderate size. They can be smaller, or they can be much larger. So nowadays we say GIST is a type of cancer or sarcoma that arises in the gut, and we now realize that this is completely different than all other types of cancer and needs to be approached completely differently. This likely arises from these interstitial cells of Cajal, which are the pacemaker cells, and again, it gives them some unusual properties not seen in other cancers. Most commonly, they arise in the stomach, but it can be small bowel or large bowel. And you know, when we first got into this business 2000, since there was no definition of GIST prior to 2000, we actually didn't know how many people with GIST there were in the United States. We were trying to do a clinical study, and we thought there might be only 300 people in the United States that had GIST. But it turns out that it's more common than that, although it's rather uncommon. So there's about 5,000 to 6,000 new cases per year, which kind of puts it in the same frequency as Hodgkin's disease or CML or a few other diseases. So uncommon, but not that rare. And again, part of the story was that prior to the year 2000, not only did we not know how to diagnose this, we didn't actually have any treatments that work. So how do people present? Sometimes we find them just by routine accident. So you're having some stomach pain or not. Somebody looks in your stomach because they think you might have an ulcer, and they may find a very small tumor, which you didn't even know that you had, or you may go to emergency room because you have a kidney stone, and they do a CT scan, and they find something that they see on the scan which had nothing to do with your kidney stone. If the tumors are bigger, then of course they can cause symptoms, and the kind of symptoms depend on where the tumor is and how large it is. So you might just feel like something's not quite right, or the tumors, if they're larger, can bleed, and so you may become anemic, or you may have signs of external blood loss, so you can see blood that you've thrown up or blood in your stool. You can be anemic if you're bleeding, and it can just produce nonspecific symptoms. So a stomach, kind of important for eating. If you have a tumor there that's taking up space, you may lose your appetite, may lose weight. That can make you tired, and so you can have sort of nonspecific complaints. Or sometimes people present with sort of a surgical emergency. The tumor actually ruptures or has a big bleed, and so patients show up in an emergency room without any knowledge they have cancer, and all of a sudden they're going to an emergency operation because of what's going on. Now, Dr. Corless will talk about this, but again, going back to my message of mutation, 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 that's our drug target. And so most GIST, about 86% of them or so, have a mutation. So we started off knowing about the KIT protein, which is over here, and there's different locations called exons, which is a location in the gene where it could be mutated. And we'll talk a little bit more about that, but depending upon the type of mutation, that can affect what dose you should be on. Or some mutations over here, like in PDGFRA, they do not even respond at all to our conventional treatments, and that's why we're doing clinical studies to understand them, or how to understand how to better treat them. So there are a lot of different mutations, but you'll notice that all of these things don't add up to 100%, which means that there are some tumors that don't have KIT or PDGFRA mutations, which we're increasingly recognizing, and now using more sophisticated testing like that developed by Dr. Corless, or that by the future Dr. Bannon in the back of the room, one of my graduate students. We can probably get up to about 98% of GIST. We can figure out what caused them, but there's still 1 or 2% of the pie that we can't figure out what caused the tumor, but we're still working on that. So we talked about this KIT thing. So what is it? So KIT is a protein. It sits both inside the cell and outside the cell. So in this is sort of the wall of the cell. This is the outside world of the cell. This is the inside world of the cell. So KIT has two functions. One is it can bind a hormone, that's on the outside, and on the inside it has an enzyme activity, which is sort of the power source. So normally the way KIT works in a non-cancer cell is it just sits there doing nothing, so it's not a problem. However, KIT can do things that are useful. So under appropriate circumstances, if your body makes a hormone called KIT ligand, then it can act sort of like a glue that brings two of these KIT molecules together. And when they're in a close approximation, then the enzyme activity becomes activated and it sends signaling, and so that causes cells to grow. So there are certain kinds of cells, like pigment cells or bone marrow cells, that depend upon the signaling, but it's normally regulated by whether the hormone is present or not. In the case of GIST with KIT mutations, the protein becomes activated without the hormone being present. So if this is the enzyme power source for the cell, instead of being sometimes on, sometimes off, it becomes always on. And that's really the driver of the tumor, and our goal of our therapy is to turn off that power supply. Now, it turns out, as I've sort of alluded to, that we started off with knowing that GIST was one thing that had KIT mutations. Now we know GIST is at least 20 different diseases, some more closely related to others. So we can look at KIT mutations, we can look at PDGFRA mutations, we can look at other mutations. All of these tumors might need a different therapeutic approach. So the drug or the treatment that we would apply can vary. That's why we need to know what you have, and Dr. Corless will talk more about this, in particular how we do the testing that results in this knowledge that allows us to figure out where you fit in this table and then go forward with therapy. So again, mutation, mutation, mutation. So why is it important? Well, this will help inform the decision whether taking Gleevec for as long as 3 years or maybe for the rest of your life after surgery will help you or not. Why take something that's not going to help you, that has some side effects, that is expensive, and if it's not going to help you, why take it at all? So knowing that mutation is important for that. In some circumstances, although this is only a couple % of the pie, you may have inherited the susceptibility to the GIST from your parents, which means that your brothers and sisters, your parents, and your children could be potentially at risk for getting GIST as well. in that system or in that situation, we can test to determine who in your family might be affected, and there's effective screening ways that could prevent them from getting GIST or other tumors. Knowing what type of mutation can determine what dose of Gleevec you should be taking, whether it's 400 a day or 800 a day. And also, and Tracy will talk more about this, increasingly because GIST is diverse and different treatments work different or the same, in order to go into an experimental drug study, we need to know what your mutation is. So when people call up all the time asking to be in a clinical study, the first question we ask, what mutation do you have? And if the answer is, I don't know, then we help you find out so that we can answer the question of whether or not we actually have a clinical study that will benefit you. Now, outside of OHSU, only about 10% of GIST patients, Life Raft, Michelle could verify this, that's still the right number?
8 to 10%.
8 to 10%. So 90 to 92% of GIST patients do not have this critical information for their doctors in order to make the best treatment decisions for them. So, you know, this is critically important. Now, in terms of what to do about it, let's talk about things that don't work. So historically, what we've done to treat GIST back in the 90s or the 80s was to treat it like a sarcoma, which is a form of cancer. We give high-dose chemotherapy. It does not work. Less than 5% of people, maybe less than 1% of people benefit from chemotherapy. So we never do that anymore. It doesn't help you in any way. Sometimes we could consider radiation treatment, but it's hard to radiate lots of sites in the abdomen, and that's where GIST usually hangs out. And so radiation doesn't really work that well either. And so really, if you have tumor that's localized, we remove it with surgery. We'll talk more about that. If it can't be removed by surgery, we do something to inhibit what's going on in the cell. So our big solution, at least in the case of KIT mutant tumor, so here we have the KIT mutant protein, it's turned on. However, we can add a drug, Imatinib or Gleevec, and now we have two other drugs we could use, and that inhibits the enzyme activity, which is the same as turning off the power supply. So we're just sort of ripping the battery out of the cell so that the cells cannot grow. And so this has tremendously impacted survival. So again, going back to the time when Mark was born in the 90s, if we treated you with chemotherapy, which didn't actually work, although we hadn't figured that out at the time, the average person with advanced GIST lived about 6 months. When we started doing our first clinical study in the year 2000 using Imatinib or Gleevec to treat GIST, we immediately kicked up survival on average from 6 months to 5 years. That was sort of a different era because people had a lot more advanced disease because they kind of had accumulated from nothing else working. Nowadays, the average patient with advanced GIST has much smaller volume of disease, so we think that the average median survival starting on frontline Gleevec is around 7 years. I should say that this was our first study from the year 2000, so we still have patients from that study who are now 16 and a half years into their treatment who are still doing well, still on Gleevec. So we don't know what the upper limit is of how long treatment can be effective, because every day that they take the drug, they're setting sort of a new world record for themselves and for everybody else as well. So this is the diagram of how we treat GIST. So we're going to kind of walk through it step by step to sort of talk about all the sort of possibilities. So the first thing we'll talk about is primary localized disease, which is we found a GIST, it's in your stomach or your small bowel, it's not anywhere else, so what do we do? So if we can remove it, we remove it with surgery, and this can be curative, although the risk of recurrence is variable. So in some people, we remove a tumor and we perform a risk estimation, and Dr. Corless will talk more about it, and we will say, you have a 2% chance or less that the tumor will come back, don't worry about it, we'll do some scans, we're not going to treat you. There's, you essentially have a 98% chance of being cured. Other patients have a much higher risk of recurrence, which could be 90 or 95 or closer to 100%, and those we are much more worried about recurrence, and then we do things after surgery, which we'll talk about, to prevent recurrence. But if we can remove it in one fell swoop, we will do so. Now, sometimes we see a patient, they just have one tumor, but we don't think we can remove it when we start. And so in that case, we do something that's called neoadjuvant therapy. So neoadjuvant means before surgery. So we're giving a medical treatment before surgery to shrink the tumor to the point where we think the surgery could be more successful. Now, most cases we don't need to do this because we can just remove the tumor now and the surgery remains the same. But there are sort of some tricky locations where that's not possible. So one case is the duodenum, which is the first part of the small bowel after the stomach. It's kind of a tricky location. You got the stomach coming in, you've got the pancreas coming in, and you've got the bile duct coming in, and they're all in about that much space. And if the tumor is kind of in the wrong location there, or it's too big, then we have to take out the pancreas, take out your gallbladder, reconnect all the bile wiring, and rewire your stomach. So it's kind of a big operation. It's called a Whipple operation. If we can shrink your tumor with a pill and make it so we don't have to remove all those normal organs, we will try to do that. Another situation is when your tumor is in your rectum or very low in your colon, and we don't think we can remove it without causing the need for a colostomy, then we will treat patients with Gleevec to try to shrink their tumor to avoid the colostomy. Or there are situations in which the tumor is so big we don't think we can take it out because we don't even know how to plan the operation. So this is an example of that. So I guess I should use the pointer. So basically we're looking at the CT scan. So here's the liver. All of this is a ginormous gist. Okay. So this is the normal liver. This is the pelvis, this is the bladder being squashed. All of this is a ginormous tumor. So we can't do an operation on this because we don't even know what it's attached to or how we could lift it all out with having it fall apart. So in this person, rather than proceed with surgery, we've elected to treat with Gleevec. So 6 months later, this is the current size of the tumor. Now you can see the bladder has re-expanded and all of normal small intestine and stomach can sort of compete for space. So now we can figure out that this tumor is coming off one of these loops of small bowel, and we can do an operation with minor removal of normal organ tissue and without any risk of tumor rupture. So in some cases, if your tumor is not resectable, we can make it resectable by treating you. The next question is, after we do surgery, do we need to do anything else? And so that depends on how risky it is that your tumor is going to recur. So again, Dr. Corless will talk more about it, but there's 4 things we think about. One is location. So the stomach is a better risk site than any place else in your abdomen. So everything else being equal, if it's in the stomach, it's less risky than if it's in your small bowel or anywhere else. Tumor size. As you might imagine, having a big tumor like I showed you is much more risky for recurrence than a 1 centimeter tumor, which has probably a less than 1% chance of recurrence. There's also something called the mitotic index, which Dr. Corless will talk more about, but it's sort of a growth rate estimate of it. And finally, the tumor, if it ruptured spontaneously or ruptured during your surgery, which means it kind of spilled cells everywhere, then that's a high risk for recurrence. So there's different ways to do this, and Dr. Corless shows a different way, which is a table. I kind of like this approach, which is if you're hiking, this is sort of like a topographical map, but instead of showing elevation, we're showing risk of recurrence. And the risk scale is down here, so sort of red hot means really high recurrence, and cold blue means not much chance of recurrence. And so we can look at where your tumor is, we can see whether it's ruptured, we can look at the size of the tumor and the mitotic index, and we can come up with an estimate of risk. So for some patients after surgery, if your risk is low, we'll not do anything but observation. If your risk is higher, and again, what does higher mean? That's sort of in the eye of the beholder, but generally above a 20 to 25% risk of recurrence, we would talk about taking Gleevec after surgery to prevent recurrence. So our knowledge of giving what's called adjuvant, which means after surgery, adding to surgery, is based on clinical studies, which were conducted. And this was a straightforward question. We previously had decided that giving Gleevec for 1 year was better than not giving it at all. This was a duration question. Is it better to take it for 3 years than it is to take it for 1 year? And so patients were part of a clinical study and Tracy will talk a little bit more about clinical studies, but people sign up for it. You know, it's optional, it's not mandatory, and in this case, they're randomly assigned to either get standard treatment, which at that time, which was 1 year of treatment, versus 3 years of treatment, and then we looked at what happened. Now, it's important to remember not every GIST in the world was eligible for this study, so it's what I call the rule of 10. So if your tumor was bigger than 10 centimeters, or your mitotic index, which Dr. Corless will describe further, was more than 10, or your tumor was more than 5 and your mitotic index was more than, should be 5, not 10. So 10, 10, or 5 plus 5 gives you 10. Any of those were high risk, and they were eligible for the study. Patients with smaller tumors or lower mitotic index were not eligible for the study because it was viewed that their risk of recurrence was low enough that you didn't need to do this, or if your tumor ruptured. And so this is looking at what happens to people after the study. So on the left is recurrence-free. So at the start, no one has cancer because they all had it removed, and we follow them over time. If your cancer recurs, then the curve goes down. So if it went to 0, that meant that everybody recurred. This is looking at 3 years versus 1 year. So you can see that the patients with 3 years had less recurrence. And if you look at survival, again, everybody's alive at the beginning. More people are alive kind of at every point in time, especially out 5 or 7 or 8 or 9 years later, if you took 3 years versus 1 year. So this is sort of our basis for thinking that for patients with high-risk tumors, after surgery, you should take Gleevec for 3 years. There's some thought, and we've done clinical studies looking at longer durations, 5 years or maybe even forever. We don't have any data from that. So currently, if you have a high-risk tumor, 3 years would be the standard therapy. So what if your tumor has spread? So now we're over on this part of the chart where you have what's called metastatic disease, tumor that spread. In that case, we would give you Imatinib. Again, mutation, mutation, mutation. So depending on what type of mutation you have, you should take different doses. Most people should take the standard dose of Imatinib of 400 milligrams per day. However, if you have the KIT exon 9 mutation shown in red, which is about 9 to 10% of patients with GIST, then you should take more Gleevec, 800 milligrams, because we know from systematic studies that people will do better to take the higher dose. So again, if your oncologist doesn't know your mutation and gives you 400 and you have an exon 9 because nobody tested for it, you're less likely to have a good outcome than if you were taking the correct dose. We'll say no. So again, this sort of emphasizes the point that here is a patient who had a very large tumor in their pelvis. Kind of everything you see that's not real white, which is the pelvic bone, is tumor. And after 6 months of Gleevec, there's a big response. But there's still a little bit of tumor left. So these are the cells that are hibernating potentially. Some of it's scar, some of it's hibernating. But this is the reason why you can't stop taking Gleevec. Because those cells will come back to life and it could grow and be as big as it was before. So, Gleevec can work for really long periods of time. For some patients that we have, they're 16 and a half years in and going. But unfortunately, in some people, the tumors can become resistant. So the tumors can start growing despite the fact that you're doing everything right, you're taking Gleevec, you have the right dose, but the tumor changes. And we call that resistance. So there's sort of two scenarios where that happens. One is if it happens right away, that's usually due to something about your tumor that we could know at the baseline. So it was a KIT exon 9, but we gave you the wrong dose. It was something called PDGFR D842V, which is not going to respond to Gleevec at all. And we have a clinical study of a new drug for that, but Gleevec is not going to work. Or you have wild-type GIST, which Dr. Corless will talk about, but wild-type GIST is about 15 different types of things, and so a lot of them, or maybe most of them, won't respond to Gleevec. But what can happen in patients who have taken Gleevec for long periods of time, like years or even a decade, that you can have the tumor start growing again even though it has been controlled for a long period of time. And this is usually due to the fact that the tumor is mutated and you've got additional mutations. So this is the KIT protein, and you sometimes see these on pathology reports. And so what can happen is that additional mutations develop because the tumor can mutate. And remember, we saw the movie that the Gleevec had a wiggle in, so it's like a lock and a key. And some of you probably have heard me say this before in clinic. So that's how it works. But the tumor, when it mutates the protein, it changes the lock. The key won't go in no matter how much you take it. However, we have different kinds of keys that will fit in, and so we can switch drugs and overcome resistance. But these are the things that we're combating, which is not a infinite number of problems, but it's the same problems over and over that we battle with our drugs. So what do we do about it? Sometimes we increase the dose of Gleevec. That sometimes works. If it's only one spot, or we think we could remove the spots that are resistant, we remove them. We can go to a second-line treatment with sunitinib or Sutent. If that doesn't work, we can go to a third-line treatment with regorafenib or Stivarga. And again, it's all about the mutations. So even when we know what's going on with the mutations, Sutent, shown green, will work on these mutations, but not on these mutations. So again, it gets more complicated after Gleevec because not all the tumors we're battling are the same, and so sometimes we give a drug, and some tumors shrink, some tumors grow, and some tumors stay the same, and we're trying to balance out, do we think that the person overall is deriving benefit or not. After regorafenib, then there's no approved agent, so you really should be seeing a GIST expert, and the Life Raft group can help with that, helping you find patient doctor lists or referrals, because the average oncologist probably has 0 to 2 GIST patients, and you never want to be in a situation where you're the only patient with a problem that your doctor's ever had or ever will see. So you want to be seen by someone who's expert. There's other things we can try, and we can battle your insurance companies or battle with patient assistance programs to try them. So these are approved drugs for other cancers that can sometimes benefit. Tracy will talk about the role of clinical studies, which is, you're in the land of the unknown, so oftentimes, you want to consider these clinical studies, and if you think about it, Gleevec was a clinical study at some point, and people had to sign up for that. Or we sometimes think about surgery if that's possible. So to summarize, GIST is diverse, mutation, mutation, mutation. We need to know what's going on in your tumor. After surgery, Gleevec can help. But if you have advanced GIST, the tumors can become resistant, and that is a problem. We have people in the back of the room and smart people around the world working on this problem, so we hope to do better, and we have clinical studies of some new drugs which are showing promise, and Tracy will talk about that. And again, thank you for your support, Life Raft Group, for referrals, and also for financial support of our research, and all of the patients who have participated in our clinical studies. Thank you. The world-famous Dr. Corless is going to now talk more about pathology and mutation testing. Mutation, mutation, mutation. Right?
Thank you, Mike. I appreciate the opportunity to be here tonight. How do I get to my show? Okay. Yeah. So, Mike and I have been working together on GI stromal tumors since 1999 when he walked into my lab and he said, "Have you ever heard of a GI stromal tumor?" And I was one of the few people around who had, and so he said, "Can you get me some?" The pieces left over from surgery. And I said, "Yeah, we can do that." And so we started working together. And I think because we've been working together so long, there's a lot of parallel thinking going on, which means that there's some similarity between his slides and mine. But as we say in medical school, repetition is learning, right? So hopefully this won't get too boring. We'll try to get through it. Do I need to put it in? Okay. Oh, it's recording, right? Okay. Great, thank you. So let's go ahead and move forward into the first slide. So Mike presented to you the basic distribution of GIST. 60% presenting in the stomach, 25% in the small bowel, and then we have these other locations. 5,000 new cases per year in the United States. That's a lot when you think about 5,000 individual patients.
But compared to colon cancer, 140,000 new cases each year, or lung cancer, over 200,000 each year, it's kind of a small disease. And that's why it hasn't necessarily received as much attention as it needed to in the past. What we see in the microscope as pathologists varies a lot. And that was one of the reasons why originally this tumor was mixed up with a lot of other tumors, because all we had was the microscope to look at. And you can see from these images, all of which are GIST, that they look different. And they get mixed up with other cancers. And that's why the identification of the KIT protein and being able to stain for that allowed us to begin to identify these things much more accurately. And here's an example of, again, that staining. So we're looking at the brown stain for KIT in 3 of the panels. And then there's another marker that we sometimes use called DOG1, D-O-G-1. Interesting story. That was discovered at Stanford. And it was called Discovered on GIST. That's why it was called DOG1. But the real reason is it's because our friend Matt Vanderine at Stanford really loved his dog. True story. Okay. So DOG1 is the other marker. And between these two markers, we can nail down the diagnosis of GIST. So when we first started working in this field, people were still mistaking these tumors for all kinds of other things. Today, I would say we're at 99.9% accuracy here in the U.S. So where did GIST come from? Mike introduced the concept of the interstitial cells of Cajal, the gut pacemaker cells. And this is a cartoon that illustrates for you in yellow on the left this tiny network of cells that lie within the muscular layer surrounding the stomach as well as the rest of the intestine. And there are a lot of similarities between these ICC cells and GISTs. They both express KIT, they both express DOG1, they express ETB1, and there are many other parallels as well. So we're pretty convinced that GI stromal tumors come from that population of cells, even though otherwise you wouldn't pay them much mind. But there's even more compelling data. So a number of years ago, and this has now been reproduced by several labs, a mouse model of GIST was made. How was that done? Take a mutant form of KIT, introduce it into the mouse genome, and then now the mouse has an abnormal KIT protein, just like we see in human GIST. And the result of that in the mouse GI tract is an overgrowth of those ICC cells. So all you have to do is change one molecule, make one mutation in it, and suddenly all the ICC cells begin to grow in an abnormal mechanism, in an abnormal pattern. And in fact, they even form nodules, like you see here on the right, down here in the colon, that look a lot like GIST in humans. Okay. All right, so that's pretty compelling data that ICC cells and GIST are related. But where do they come from in the general population? And this was a bit of a surprise, but we noticed early on, and other groups did as well, that occasionally we would run into something that was a GIST, but boy, it was small. And it wasn't something that was picked up clinically, it was incidental. Because the stomach was removed for an ulcer or for a cancer of another type. But nearby, we find a little, like a pebble, like 1 centimeter, less than 1 centimeter in size. Boy, it stains for KIT, it looks like a GIST. And in fact, it is a baby GIST. They even have mutations in them. And how common are these? Several studies have now established that in adult population, between 10 and 30% of patients in the general population have one or more of these microGISTs in their stomach. So, who has one? There's a few more around here in the audience, okay? All right, what that tells us is that there's a lot of these benign GISTs in the general population. And most of them never bother us. And there's a parallel here if you think about prostate cancer. There's a lot of prostate cancer in the older male population here in the U.S. Most of it never bothers us. Okay, so if we have several million of microGISTs in the general population, only 5,000 per year actually become something we regard as clinically important. And out of that, maybe 2,000 actually become malignant. Okay. There then are other factors that turn these tumors into something that's malignant. It's not just the KIT mutation or PDGF receptor alpha mutation. That's the starting point, that gets those ICC cells growing in an abnormal way and forming these little nodules. But beyond that, we have to have other chromosomal events that allow the tumor to become something much more aggressive and much more dangerous. We have, and other groups have been studying these events. They're beginning to be laid out and understood, and to suffice it to say, that probably takes about 6 steps, or if not more, before something goes from a completely benign GIST to an overtly malignant GIST. So Mike showed this slide, and I just once more want to emphasize that there is this interesting distribution of mutations in both KIT, and we've been emphasizing KIT because that's the big player. 75% of GIST have mutations in KIT, but also a sister molecule called PDGF receptor alpha. About 10% of GI stromal tumors have mutations in these, and they're mutually exclusive. It's either one or the other or neither. And the neither is the remaining population of what we call wild-type GISTs. Sometimes I like to think of them as wild card GISTs because we used to not understand how these things were growing. Recently, there's been an uncovering of a number of other kinds of mutations hiding in that wild-type population. And so, as Mike emphasized, we're now at the point where we can identify the basic cause for about 97–98% of GISTs. Okay. So Mike also showed how the mutant forms of KIT, or PDGFRA, the dominant players in most GISTs, will cause these signaling events. We talked about those little lightning rods going on. Diagrammatically, or cartoonish, this is what happens going on in terms of that lightning signal being transmitted from the surface of the cell where the KIT molecules acting down to the nucleus, where we see changes in cell, in gene regulation that tell the cell to grow. And by adding a drug like Imatinib, it goes in and basically blocks that signaling and turns it all off very nicely. Okay. Once more, exon 11 mutant tumors have the best overall response to Imatinib and compared to other kinds of GISTs. The exon 9 mutant tumors require a higher dose, and there is an unusual mutation in PDGFRA that we don't have a good drug for except one in clinical trials. Now, the molecular classification of GIST is even more complicated when you begin to look at the wild-type population. This is too small for those of you in the back of the room to read, and I don't want to go through it all. But suffice it to say, there are now a number of other subtypes that constitute this total of 98% that we think we can understand these days. And many of them actually have drugs available. Not Imatinib, not Sunitinib, it's going to have to be something different. But we do have drugs available for them. One of the most common, though, amongst that wild-type population are this group of tumors called SDH-deficient GIST. Okay? SDH is an enzyme, stands for succinate dehydrogenase. And these tumors are missing that enzyme. They're defective. And they are unusual in that they have a different growth pattern. They're very nodular. And when I see them on the microscope, I think to myself, that might be an SDH-deficient GIST. And I can confirm that by doing a stain for SDH. And if it's clear, there's no brown stain here, that means it's one of these kinds of tumors. They're only seen in the stomach. I don't know why, but they're only seen in the stomach. And they don't respond to Imatinib. So we have to think about other treatments for this type of GIST. It's about 12% of all GISTs. These are caused by mutations in genes SDH A, B, C, or D. Those all work together to make that complex of enzyme. And about half of these, we think, are inherited. So suddenly now we're moving from most GISTs where you don't have an inherited component to the disease to this subset of GIST where frequently there is an inherited risk called the Carney-Stratakis syndrome. Not only are you at risk for GIST, you can develop other kinds of tumors like paragangliomas and pancreatic tumors and renal tumors and so forth. Okay. And also, very strange, you can have different penetrants. What does that mean? One patient in a family may have a mutation and develop a GIST. The father may have died of a paraganglioma, and the brother, who also has a mutation, age 60, no tumor at all. We don't know why. So there's a lot of mystery still that we have in this disease that we need to come to understand. Now, because Mike keeps emphasizing mutations, we got to go looking for them. How do we do that? We do that with what's called DNA sequencing. meaning we're reading the DNA, and determining the letters. And remember, DNA is pretty simple. It's made up of 4 letters, A, T, C, or G. And it's the sequence of those that determines what proteins are made and how the cell functions. So in order to read the DNA, we have to first unravel it, and then we create copies of it in the test tube, and that allows us to read it while we're copying it. But every time I see this cartoon, it always reminds me of this cartoon. So the son goes up to mom and he says, so can I go down to the lake? And mom says, well, let me go see what your dad thinks. And he says, well, remind him I could really use some relaxation. So when mom checks with dad, his answer is, if he relaxes anymore, his DNA will begin to unwind. All right. So it kind of reminds me of my brother, but we won't go there. Okay, so we want to copy the DNA, and in the process, we're going to read that. And these days, we've been able to see the benefit of a tremendous revolution in DNA sequencing technology. The original sequencing technology looked like this, a bunch of colored lines going up and down, and the different colors meant the different letters. And you would read it, but it was messy, and it was painfully labor-intensive to create the sequence, and you couldn't read very much. You can only read a small piece at a time. Nowadays, we do sequencing that is basically digital. Out comes the straight, the letters just like this, and you can see the red T's meaning mutation versus the normal A's, and you can even determine quantitatively how much mutation is there versus how much normal there is. Okay. In my mind, this is very similar to the comparison between the TV that we had in my house when I was a kid. Yep, black and white, analog. You had to go over and turn the channel by yourself. Okay. Didn't have a clicker. Okay. We did have one of these, believe me, I'm that old. And today, right? Hundreds of digital channels. Well, our sequencers have millions of digital channels for doing sequencing simultaneously across many different genes at a time. And it's very, very powerful technology. We have actually a couple of different instruments in the lab that allow us to do this. One of them is this box right here. We have a couple of these, and basically the DNA is spread out on what is essentially a glass slide, looks like a microscope slide. And then there's a high-resolution camera that's following the sequence, and each of the nucleotides, A, C, T, and G, has a different color, and the camera's just watching the change in color as the DNA is being created. It's very cool. Okay. And it also turns out very large files. So if you think about downloading a movie, this is about 100 movies worth just to do one run of sequencing. The other sequencer, and we have 3 of these in our lab, is one where we do the DNA sequencing literally on the surface of a microprocessor, a little computer chip. Very cool technology again. The DNA is put on the surface of these little spheres. Those spheres are squirted on top of the chip. It sits down, and there's a lot of chemistry going on inside, and from that we can get the sequence out. These are the kinds of things, I have to tell you, these are all only a few years old. This one was first demonstrated to me in 2010, not very long ago. And when the rep came from the company and said, this is our new technology and this is how it works, I said, no way. I didn't believe it. I was like, that's not going to happen. But we've been using it very successfully to sequence GIST and many other tumors. It really does work. We've created a panel of genes. So when we want to really dig deep into a tumor because it doesn't have a straightforward KIT mutation or PDGFRA mutation, this is what we go after. We sequence all of these genes simultaneously in one run on one of those chips. And that tells us pretty quickly if there's another kind of mutation, including one of the SDH genes, that we want to know about. Now I want to show you, in a sense, what it's like then to try to interpret DNA sequence, okay? And we're going to use an example from the Bible, all right? I hope this is visible to you. There is this segment of a Bible that was published in the 1600s, King James Version, and it says here, thou shalt not kill, thou shalt not steal, thou shalt not bear false witness. Sound pretty familiar? There's one problem. Right here it says, thou shalt commit adultery. Whoops. Whoops, wait a minute. It That's not supposed to happen, okay? So this was known as the Wicked Bible, 1632 edition. Once the error was discovered, they went around the countryside trying to grab all those editions and burn them. But as it turns out, some of them survived, and we have this as an example of a printing error, right? What happened was there's a deletion of one word, the word not, and we see that exact same thing in the DNA sequence of GI stromal tumors, okay? Here's the KIT gene. We're looking at a series of amino acids which are encoded by these nucleotides from the DNA. And right here, there are 3 of them missing, which means that aspartic acid in the KIT protein is gone. And that's all you need. That single change turns the KIT molecule on and makes it a very powerful driver for the growth, the initial growth of a GI stromal tumor. One more example is another one from the Bible, 1795 edition. We have, let the children first be filled, became let the children first be killed. Ouch. Okay. So that is an example not of a dilution, but of a substitution, right? We substituted one letter for another. We see exactly that thing in KIT as well as in other tumors, the BRAF gene. We're replacing this T with an A, and that's all you need to do to go from valine to glutamate, and that means this is turned on and is driving the growth of a melanoma in this case, but we see similar mutations in KIT as well. Okay, so it gives you an idea of the kinds of changes, very subtle changes in the DNA code that lead to these very considerable consequences that we see in tumor growth. Okay, now, Mike wanted me to discuss a little bit about how we assess how actively growing a tumor is and what the prognosis is. Okay. First, I want to show you this study that was done prospectively, meaning from this period forward for a period of time, we're going to see in this particular region of France, the Rhone-Alpes region of France, how many GISTs are diagnosed. And this was over a 2-year period, and the answer was 115 GISTs were diagnosed. Okay, so now you're following it, not looking backwards in time, but just in real time over a 2-year period. How many new tumors? 115, and out of those, 88% had not spread. So that's kind of good news, because those 88% of patients theoretically are cured by surgery. Okay, only 12% actually had metastatic disease when they walked in the door. Okay, so that's great. However, we then need to look at the risk of recurrence, and I'll show you how we do that in a moment. But the risk, as you see, varies considerably from about 1/3 or a little bit more than 1/3 that are very low risk, probably don't need to take Gleevec at all, to some that are in the middle region, and then more than 1/4 that are high risk, and they definitely need to be taking Gleevec. Okay, so what that means is that even though many of these patients are going to be cured by surgery, a considerable fraction are going to have to consider the use of Gleevec after the surgery. So how do we assess it? Mike introduced 4 factors that we look at: location, size, and mitotic index. Sorry, 3 main factors, tumor rupture being the 4th one. The 3 main factors, and of those, mitotic index is the most important. Now, what does that mean? What is a mitotic index? This is a diagram reminding you from your biology days in high school, okay, where we're taking a cell through cell division and dividing it up into 2 new daughter cells. And you'll remember that during that process, the chromosomes have been replicated, and at one point they all get lined up because you've got 2 copies, you get 1 copy of each on each side, and they get pulled apart into the daughter cells. This is called mitosis. This process is dividing the chromosomes up into the 2 new cells. That's mitosis. Okay? So we can look for mitosis on the microscope. And if we look at very, very high power, this is what it looks like. So that's exactly the diagram I showed you just a moment ago, right here, like that. That's what we're seeing on the microscope right here. Okay. Now, if we go at lower power, because I don't want to spend all day looking at that, that's kind of like trying to mow, a gigantic lawn with a teeny little lawnmower. So instead, I'm going to back off to a lower power, and I still can see them. And there they are at lower power. Here you can see them dividing up chromosomes and these ones are still lined up together. And so these are examples of the mitotic figures that we look for. And of course, by definition, when we see those are dividing cells. The more we see, the more the tumor is dividing. Very simple. So all I have to do is cruise around at this power on my microscope, about 25 different areas of the tumor, and then add up all of the mitoses that I see. Hopefully it's only 1 or 2, but if it's more than 5, it's a sign that there could be a problem and that this tumor might come back. So we combine all this information together. The mitotic index, by far the most important, but we also factor in the size of the tumor, if it's 2 or 2 to 5, or it's greater than 5 centimeters or greater than 10. And then the location. Mike talked about how the gastric tumors have a better prognosis overall than other tumors, and that's borne out in these statistics here. These, by the way, this was put together by a couple of pathologists working back in Washington, D.C., from old cases before Gleevec was invented, and it was a heroic effort. This is based on over 1,000 patients with long-term follow-up. And by getting all that information together, we could generate this chart, and that's what allows us to fairly accurately predict what is the risk for a patient, and they can decide whether to take the drug. Okay. So, just to summarize then, what we've talked about is that GIST are a family of tumors arising from mutations in a number of different genes, not just KIT. Most GISTs probably come from these little microtumors that are common in the population, but you have to have other kinds of events genetically that allow them to become malignant. We talked about how next-generation sequencing is very helpful if we're going to do deep molecular subtyping of these tumors, identifying unusual mutations. And finally, how our mitotic index is the most important, but we have these other factors that we need to look at in order to judge whether a tumor is going to come back. So with that, I'm going to end, and I'll turn the podium over to Tracy. There you go. Yeah, I don't know why that's going to go.
Okay.
Oh, yeah. Yeah, you can click it.
Excellent.
Find that, yeah.
Okay. The green, I assume.
Okay.
Okay, so my talk is going to be a lot less scientific than the two you've seen already. I don't have any gross pictures. I don't have any of the cool protein videos coming together. I kind of want to talk more about, for those of you who don't know anything about medicine or, GIST in particular, hopefully you've learned a lot tonight, but I want to talk to you a little bit on a simpler level. How to find and participate in a clinical trial really is a lot about protocol, where to go, and that kind of thing. So we're going to get into kind of some logistics about that. Most of the trials that we run here and kind of the population I'm talking about are the people that have gone through Gleevec for the most part, maybe failed Sutent, maybe failed regorafenib. You start to get kind of down the line in your head, we want you to start thinking about a clinical trial, and that's where centers like ours will come into play. It's not 100% true. We do run clinical trials for earlier, people who are just starting on Gleevec and that kind of thing, but this would typically be for people who start to fail treatments down the road.
It's not working.
There we go. So again, rare disease. My comparator here was the breast cancer population. Lots and lots of cases. So just is relatively rare in the cancer world. If you're going to think about where to go to express interest in participating in a clinical trial, The sites around the country that we work most often with are Dana-Farber in Boston, Fox Chase is in Philadelphia, MD Anderson is obviously a huge cancer center, and then Memorial Sloan Kettering in New York. There are obviously other sites around the country that are doing GIST trials, but these are kind of the biggies. You notice, and for the most part, we're the only one on the West Coast. Stanford does clinical trials. You know, there are others, but if you're talking about, again, big centers with specialty sites, these are going to be the ones that you're looking for. And that's okay. We do this every day. And I put this slide here because I wanted to show people where people come from to see us. It's not just an Oregon thing. I'd say 90% of the people that we treat live outside of the state of Oregon. We talk about a rare disease, and then you're talking about a clinical trial population. We really do have people from all over the country. We had a patient last year from Nigeria, okay, and he came once a month for about 2 years, something like that. He came once a month. I will mention that most of the time our trials, we try really hard to negotiate budgets so that we can pay for travel for people to come. So don't assume that just because we live on the west, you know, that we're on the West Coast, that it would be impossible for you to get here. We will be happy to talk to you about how we can help you pay for that. But before we get into a clinical trial, let's talk a little bit about what are the phases of research. The first phase, phase 1, clinical trials, are literally the first time in humans. So they've often done a lot of research in the lab. They've done research with animals, mice, dogs, primates, but has never been given to a human for the most part. So if you're entering a phase 1 trial, the goal of that trial, of course we hope it works and it helps you, but literally the goal of a phase 1 clinical trial is to find out what dose we should be giving and is this safe to give. And so because of that, it goes very slowly. We're not going to just throw a new drug to humans and give it to 100 people and see what happens. It literally goes 3 people at a time, and then we wait a month. And then it goes 3 people again, and we wait a month. And so for Dr. Heinrich and I, who have ants in our pants, it is very slow going. And so we have a couple trials that I'll talk about at the end here that are phase 1. And so you may call me and say, I want to be on this clinical trial, and I'm going to say, okay, we're going to put you on a list because it's a phase 1, and we are just, it's like they have to hold us back because we want to get going, but we have to just wait and make sure that we get to the right dose. So phase 1, small numbers of people looking for the right dose and checking safety. Phase 2, so now we know, okay, we've settled on our dose. Now we want to know what happens with that dose. We'll continue to follow side effects and safety, but we want to know, does it work? Does it work for GIST? Does it work for the other cancers that maybe it touched in the phase 1 study? And then phase 3, larger groups of people. And we'll take, you know, the study medication versus what is already approved, and we'll compare them head to head. So we think this is the best treatment that we have, but does this new thing work better? And then phase 4 studies, we don't really do here. Those would be sort of after the fact. The FDA generally approves a drug already, but they might continue to follow data just to see if when you give it to tens of thousands of patients, does it develop some sort of side effect that we just didn't see in the smaller numbers. Okay, questions, by the way, anyone, stop, ask, please. So what to do before you need a clinical trial? Basics, just for anybody who's venturing down this road of what do I do now that I have this diagnosis? Check with your insurance. It's gotten better, and actually just this week I got an update. The state of Oregon has now jumped on board. Federally, the Affordable Care Act now allows patients to participate in a clinical trial without any sort of discrimination, and Oregon now follows that guide. There was an issue with some of our state Medicaid plans, but that's now been fixed. So when you're asking about insurance, people, you know, you're looking around at your job or trying to figure out what Medicare supplement to get, make sure you ask the question, you know, is it going to be okay for me to participate in a clinical trial? Clinical trials do not pay for everything. Some people come to me and just assume that they're going to, you know, hand me their bills and we're going to pay all of it. That's not true. 95% of what happens still goes to your insurance company. Most of the time the drugs are what's provided, and then maybe there'll be some study parts, things on the side that are paid for by the clinical trial. So it's really important to have supplemental insurance.
I talked about the exceptions.
Here we are with the mutation again. As we've talked about, the most common piece of the pie on my slide here is the KIT mutations, PDGF being sort of the second lump, and then there's the wild type. When you call me, and my number and all that is at the end here, if you're a patient who calls me, this is going to be one of my first questions, kind of when were you diagnosed, and then what is your mutation? And the reason, as Dr. Heinrich alluded to, is often we will have clinical trials literally that will say, you know, the only patients that are eligible for this trial must have KIT exon 11. Those are the only people that we're going to try this drug on because we've looked at it in a lab and that appears to be the group that it works for. And so there'd be sort of no point in giving it to a wild type patient. Or vice versa. We had a study just a year or so, or 2 years ago, where it was only for wild type patients. So when you call, it's really, really important to know that mutation. Currently, our studies are for all comers, so we don't have a study in many ways that is only for a certain disease. So it can work both ways. I still need to know your mutation if you call me, and we encourage you, obviously, we've talked about it a lot tonight, to know your own mutations, but it helps us kind of put you into a bucket of what we might offer and what clinical trials might be available for you. We talked a little bit about wild type. Tissue mutation testing sort of as soon as possible. As soon as they take anything out, you don't need it anymore. Get it tested for mutation with your first surgery or your first biopsy. At a minimum, if you start on Gleevec and then you fail Gleevec, we'd encourage you at that point to start to look at what your mutation testing would be. And then people always say, how do I get that done? What do I do? I send a lot of emails to Dr. Corless that say, you know, I've got a patient calling me from Tennessee and they don't know how to get their mutation testing. And we will connect you with people who do that. I will work with your local doctor's office. We do this all the time. People send tissue to us to be tested. We can help you figure out somewhere closer that might do tissue testing and mutation testing. But we do a lot of it here, and we get things sent from all over the country. Did I go backwards? No. Then the other thing I want you to do at home before you even get too far into this, you're diagnosed, you're going to figure out your mutation, and then the better records you can keep, the better it will help people down the road. When you call me, again, I'm going to ask, what's your date of diagnosis? Please, please, please keep a copy of all your pathology reports. So they might just do one. But if you're someone who failed some drugs down the road, they might go back and do additional biopsies. Maybe you have 2 surgeries, and they're going to be at 2 different pathology reports. Hang on to all of those and keep track of when did you have your surgery, where was it, and then the pathology report. We will often go back and get that tissue and have to send that in. So the more details you can keep a little gist file for yourself. It's a good idea. And then the treatments you've been on and what date. So I took Gleevec from August 2010 to September of 2012, and I stopped it because it stopped working. And then they started me on Sutin, and I stopped it because my blood pressure was too high. Kind of keep a running tally of what you've been on and when. We will ask you those questions because those are the things we get asked for clinical trials. They want to know, this study is for people who have failed Gleevec and Sutin. And so I have to know, when were you on those? How long ago? All those kind of questions. So the more detail you can keep track of, it will help you down the road. Sounds like a big job, but it's a good job for spouses. A lot of times family members are good at kind of these details. How do you find a clinical trial? There are 2 websites that we use and I recommend to families, molecularmatch.com and clinicaltrials.gov. I'm not going to go into them here just in the nature of the fact that we're running a little bit later this evening. But please feel free to fiddle around a little bit with these websites. They're really, really user-friendly. You go into both of them and it just literally says, enter the type of cancer that you have. And you type in, gastrointestinal stromal tumor or GIST, and it will pull them up for you. And then it will say, here are a list of the open clinical trials. You can click on where are those trials open, who's running them, and it will give you a list of the sites around the country that have the trial open. Many of them are filtered. The molecular match site filters by mutation, so if you know your mutation, you can click your mutation, and it will tell you what studies would be open for your mutation. It's really, really user-friendly. And then at the last part, it will link you to whatever site is open and the name of the person at that site who is running that trial or who is a contact for that trial. So it will help you get immediately to the whatever center you'd be interested in. And then just quickly, what do we have coming here, or currently have open here? Our first trial, BLU 285, is a phase 1 trial using a KIT inhibitor that literally, when we gave it to our first patient, as I mentioned, no one in the world had ever taken it. So we put the first 3 people in the world on this drug and kind of crossed our fingers. It's an oral medicine taken daily. This is one that we're having a hard time getting enough slots because we have so many people we want to give this to. It appears to be more active for the PDGFRA group. So those are kind of the patients that we are looking for at this point, but it's not been at all finalized that it is more or less effective with other mutations. It just appears early on that maybe the PDGFRA D842V group is responding a little better to this. So especially people with that mutation, this would be something that we would recommend. Again, phase 1, so there's going to be lists around the country of people trying to jockey for spots. But we do have this study open here at OHSU. Once the study finds the dose, the trial will open up a little bit more, so there will be more opportunities for people to go on this study. So don't give up on this one early if you don't get a spot, somewhere because it's kind of limited numbers. The other study we are running that's going to be literally next week, we're getting our initiation visit underway, is a drug called KTN. I can't remember the numbers. I should have written them down. It doesn't matter. But this one is an IV drug. It works very differently from the pictures we saw up here. And I'll have to have Dr. Heinrich make you some models and pictures for how this one works. But it's an antibody that basically blocks KIT activation, and so he kind of describes it as it's very similar to a drug called Herceptin that's being used for breast cancer, for those of you who maybe have heard of that. Just a little different modeling. Again, it's an IV medication, so it would be something we give it every 3 weeks. You'd be at OHSU every 3 weeks for an IV treatment. So very different from our traditional treatments for GIST in that, most of them are oral drugs that you take every day. So this one is literally opening hot off the press next week. Next Monday we have our visit. So this would be open, and this is open for all mutations. And then another one coming probably next summer, late summer, early fall, is SARC-029, oral drugs, 2 combo drugs called Imatinib and Pathology, work very differently. And so they're going to be working in simultaneously. So you'd take 2 pills on this one, 2 different pills. All comers, if I remember, mutation-wise. I think so.
Third line, maybe?
And that one is only for people who have, you have to have failed Gleevec, Sutent, and Regorafenib. Anything beyond those 3, you would not be eligible for this last trial. Okay, so it's kind of a little bit earlier in the metastatic setting for patients. But that one's coming as well, again, end of summer, early fall. And then the last slide, I just here have my contact info, and feel free to share this. I get calls from people, husbands, daughters, wives around the country all the time, and I'm happy to help you get in to see Dr. Heinrich in second opinion. If you're not already seeing him, I can help you navigate how to get in for a clinical trial. I can help you figure out if you're eligible for a clinical trial. So I'm happy to field questions and direct you to the right way, or get you to Dr. Corless's lab for mutation testing, whatever you might need. Okay? So thank you.
So our final speaker is going to be Michelle from Life Raft Group, who's going to talk about Life Raft.
Thank you everyone for coming tonight. I am Michelle Durborow, and I am the Patient Registry Director of the Life Raft Group. I also do not have the cool slides and the movies, but I do have a lot of things that can help patients navigate their care, especially in this rare disease called GIST. The Life Raft Group is a nonprofit organization that is focused on research, patient education, and advocacy to patients who are affected by GIST as well as their caregivers. We know that this is you just don't need a healthcare team, but you also need your family to help you navigate this disease. Our mission is to ensure the survival of GIST patients through a comprehensive approach connecting individual patients' needs, the worldwide community of GIST advocates, and the global health and research environment. In the year 2000, we call it the perfect storm. Patients were very advanced in their disease and they don't know what they have. They were told that they had either a benign GIST or they were told they had leiomyosarcoma. The founder of the Life Raft Group, his wife was literally told that she only has 24 hours to live and there is nothing else that they can do for her. And what happened was he went to the internet, found people who have the same problem, and they found that there is the kit training that could literally tell them what is wrong with them and that they can go into the very first clinical trial of the Imatinib, the STI 571 at that time. So this group of patients and caregivers, they all came together and formed the Life Raft Group. And to this day, most of these patients are living with GIST for over 16 years, 169 of them. We followed them over the years in our patient registry, and because of their contribution in terms of their data or their stories, we are now able to support more than 10,000 or so patients in the Life Raft Group. To do our mission, we focus on, as I've mentioned, we focus on key areas, research, patient education, and support and advocacy. To break down the services that we do at the Life Raft Group, in terms of advocacy, we help patients access safe and effective treatments. We do that by helping them find the best GIST expert in the world. We direct them to the right treatment, to the right clinical trials. We talk to key government officials and policymakers to help fund research for GIST. People know about a lot of cancer. GIST is the only cancer that doesn't have cancer in its name. When people ask, what do you do? I support patients with gastrointestinal stromal tumor, and I have to explain all the time that it is a cancer, because just because you don't see the word cancer doesn't mean that it is not. And there is a lot of work that needs to be done in bringing awareness to the disease. We also create regional groups in underserved areas. So we have representatives all over the United States and also all over the world. We do not mind if there is just one patient in that part of the world. We will support that patient and provide them with information and education. We also report on current trends in survival. As I've mentioned, at the core of our mission and our services, we have a patient registry where we've been monitoring patients for a very long time now, and we are now able to report on trends in terms of survival. And we are very proud to say that most of the patients who came to join the Life Raft Group, what we notice is that they tend to survive, their survival is longer because they are now informed, their caregivers are supported, and all other patients are helping each other. And In terms of research, we have a team of top GIST researchers from 9 institutions in 3 countries, and 2 of them are in this room tonight, Dr. Corless and Dr. Heinrich. We have a cutting-edge patient registry, as I've mentioned, and we also work closely with Dr. Corless in terms of our GIST collaborative tissue bank. This tissue bank helps us, again, as Dr. Heinrich said, mutation, mutation, mutation. 8 to 10% of patients in the United States know their mutation. That's very small. In the Life Raft group membership, 42% know their mutation, and that is because of the great work that Dr. Corless have done in terms of helping with our GIST collaborative tissue bank. We also provide patients with education. We have newsletters, websites, webcasts, and we also have a clinical trial database that is very GIST focused. So if you don't want to navigate all those big websites, all you need to do is go to our website. We have a very patient-friendly user interface clinical trial database. All you need to do is put in a little bit of information, and it will give you the latest clinical trials that are on for GIST. We also conduct GIST expert training class. We've written a module for patients and caregivers. When you take this class, you know more than the average patient, and probably you will be able to get a good care from your doctor because you will be able to ask the right questions because you know more about GIST. And we also have our Life Fest. We do every other year. We do a conference or a gathering of patients and caregivers from all over the world to talk about the latest research, to talk about what's happening to them, or to share their experience. It's a good way to network. It's a good way to find other patients who are like yourself and have gone this road before, and they will be able to give you some advice on how to manage and cope with GIST. We also have GIST Day of Learning. We do them all over the country, and this is one of them, and I can say one of the most successful. As I said, we have 36 leaders throughout the United States and 60 country liaisons, including several regional support groups, and we have a GIST specialist directory. If there is one thing that I always tell any patient who calls me, the very first thing I ask that patient is, are you seeing a GIST specialist right now? It will save your life. And also we do the GIST virtual tumor boards for those very difficult cases. We ask experts from all over the world from different fields of expertise to do a virtual a virtual tumor board to review the case and to make some and to give some advice on how to care for the patient. So what can I do today? Every patient that comes to the Life Raft group, they ask me, so what can I do today? Please first tell yourself that you did not do anything to yourself that caused GIST. The next thing is see a GIST specialist. As I've mentioned, it will save your life. Understand your GIST diagnosis, your risk of recurrence, your mutation, and your best treatment option. Mutation, mutation, mutation. I love that phrase, Dr. Heinrich. Join a support group like the Life Raft group. It's important to know that you are not alone. There are thousands of patients out there, and they are willing to give you the advice that you need because some of them have been through this for many, many long years. it's nice to have somebody to talk to who understands what you're going through. Participate in the Life Raft Group Patients Registry and contribute your data to just research. We have a patients registry that is now over 15 years of data collected from patients from all over the world, and it has informed a lot of patients. Sometimes we do not have the answers on what's happening in the real world until we gather all those data together and we find out what are the trends and what questions can we possibly answer. Donate your tissue to the GIST Collaborative Tissue Bank. This will greatly help accelerate research. Some of the patients do not realize that your tissue is just sitting in the pathology lab in your hospital, and after a few years, those tissues, they will probably throw out. So why not donate them to research? Support the cause by volunteering your time and talent. We do need volunteers. Right now, we are looking for a representative for Portland because our representative is overworked, and she would like to have somebody to help her. It's to provide support for patients in this part of the country. Please help in any way you can by making financial contributions to continue the research, to find the cure, and to provide support and education to patients and caregivers around the world. There is always something that we can do to help the cause in GIST. So thank you so much for coming tonight.